Hey guys, and welcome back to Now I Know. Today we are starting with one of the most requested topic, and that is the Operon system, and in this video we will look at Lac Operon. Now whether it is eukaryotic cell or a prokaryotic cell, it is not required by a cell to produce all the gene products at all the time. It is not needed for all the genes to be expressed at all the times. There are certain genes like housekeeping genes which should be expressed at all the time because we need the product. Other than that, there are some other genes which needs to be expressed only under certain circumstances. So there has to be some kind of regulation which takes care that these genes are switched off when they are not needed and they are switched on whenever they are needed. So this kind of a regulation in prokaryotes is by the Operon system. Now in prokaryotes, this regulation occurs at the transcriptional level. Now if it is at the transcriptional level in prokaryotes, now we are talking about all here is the prokaryote, right? So if it is at the transcriptional level, we either want the transcription to be off so that there is no waste of energy, and whenever the cell wants, it should be on. The transcription should go on and the protein should be produced. So if there is something which is regulating the transcription, which is inhibiting the transcription, which is keeping the genes off, such kind of a control is called negative control. Right? You get it? Negative because it is actually inhibiting the transcription, right? And on the other hand, if you want the transcription to go on, you want the transcription to occur, so you're promoting transcription or you enhance the transcription. Such kind of a control is called positive control. This is just the base I'm giving because I'm going to use these terms here and there. But so Operon can be positively controlled or negatively controlled based on what is needed by the cell. Now as we go on and see different type of Operon, you will understand which one is positive and which one is negative. Today what we are talking about is the Lac Operon, and that is negatively controlled inducible Operon. What is this long fancy name? Negatively controlled. Okay, let's look at it this way. The lac stands for, a lac comes from the lactose. It is an operon. Now I have not told you what exactly operon means. Just in a moment I will say. So this is a kind of a system that takes care of lactose metabolism in the cell, in the bacteria. Now lactose is a disaccharide. That means the cell needs enzymes to break down this lactose into glucose and galactose and use that as the carbon source. So that's a lot of work, that's a lot of energy expense. So a cell would rather go for a simpler source as glucose, which is a monosaccharide, simple source to be used. If at all in the environment, now they are bacteria, okay? These bacteria, they don't have a choice of food, right? They can't choose their food whether I want glucose today or lactose today. They have to have whatever is present in the environment. If they are in a body, whatever we consume, whatever we eat, they have to use a source from that. Or if they are in media, if you are growing them in the lab, whatever media we are growing them in, whatever source of carbon we are giving, they have to use that. They don't have a choice. So if they are in an environment where they don't have any other simpler source and they just have lactose as their carbon source, they have to break this down, right? So they are going to break it down when there is nothing else but only lactose present. So lactose is actually inducing or turning on the genes that are needed for the metabolism. So it is inducing the gene transcription over here. So that is why it is called inducible operon, and this will be crystal clear once we see the whole operon system and how it works. Just the baseline I'm giving, just the background I'm giving over here. Now let me first tell you what does this operon mean. So the operon is when you have a gene construct, when you have cluster of gene lying next to each other, they are involved in one function. Say for example, as I said, for lactose metabolism, it requires 3 enzymes. Cell needs 3 enzymes to break that down. So it requires 3 genes. So a cell would have 3 genes next to each other, this cluster of a gene that is present is under the control of one single promoter. Okay, this assembly is called operon. A cluster of genes that is together under one single promoter, that is the operon. And operon is a feature of prokaryotes. There will be different kind of operon based on what is, what are the genes that are getting expressed, and we'll see all one by one. Now, Lac Operon is the one which was first studied thoroughly by Jacques and Monod in prokaryote, of course, in the E. coli, and it is the example of negative operon. As I said, negative is because lactose is a disaccharide. Unless there is no other choice for bacteria, we don't want the genes to be expressed. So we are inhibiting the expression of these enzymes in the absence of lactose, and that is why it is negatively controlled. And only when lactose is present, there is no other choice. It will force the cell to express the gene. It will induce these genes. So it is called inducible operon. Just the few points that you should know. So presence of lactose cause the induction of operon, and absence would cause the inhibition. So it is negatively inducible operon. Now let's look at the assembly. How does it look like and what happens actually? Now looking at this diagram, don't worry. Trust me, it's very easy. Once you understand what each of this means, it's going to be easy to remember. Now it's a Lac Operon. That means it's for the lactose metabolism. Now certain things that you should remember is lactose metabolism requires 3 enzymes, and once you understand the mechanism or which enzyme does what, you will even remember this. So it requires 3 enzymes. So first thing that you should have in mind is you should have 3 genes that codes for these enzymes, right? And these are nothing but structural gene. They are called structural gene because they are coding for the enzymes needed. So there are 3 structural genes in case of Lac Operon, which is called Lac Z, Lac Y, and Lac A. Now how I used to remember this as A, Y, and Z is the first alphabet is the A. So first gene is A, and then the last 2 alphabets, that is the Y and Z. So it is A, Y, and Z. Now these 3 are called structural gene that is going to code for your protein, for your enzyme. Now these are the genes, of course it needs a promoter, so that promoter is called Lac P, or promoter for Lac genes. And between this promoter and structural gene, there is a region that is called the operator region. Okay, this is O for operator, Lac operator. Now we'll just come to this, why this operator is needed between the promoter and genes. We'll just come to it. Now it's a type of a system where it has to regulate it, right? Where it needs to inhibit the transcription of these genes in the absence of lactose. So there has to be some inhibitory gene that causes some inhibition. That is the portion called Lac I. This is Lac I. I, you can remember, stands for inhibition. That is the inhibitory gene. Now it's a gene, right? So it will code for something. It will code for some protein, and that is the inhibitory protein or inhibitor, or you can also say repressor, right? And of course you need a promoter for that. So you have promoter for inhibitory gene or I gene. Now these structural genes along with its promoter, this assembly right over here is what we call Lac Operon. Okay, this is an operon where it has a promoter and all the structural gene clustered together. And this portion where it is coding for enzyme is the coding region, and simple enough to understand. So this structural genes is the part where the coding region lies, and the rest of the part is actually involved in controlling the expression of these genes. So this is the control region. Once again, as I tell you the mechanism, the whole thing will become crystal clear. Now let me just write down what each of these means so it becomes easy for us. Alright, so P stands for promoter. Here PI is the promoter for the Lac I gene, and Lac P is the promoter for all the structural gene. The I is the inhibitor gene because it is a regulatory mechanism, so there has to be something that is actually regulating the expression, regulating the transcription, and that is the inhibitory gene or inhibitor gene that codes for a repressor protein. I'll come to this, what repressor does. O, Lac O is the operator site. Then you have like Z, Y and A. I said these are the three genes that code for three enzymes which are needed for lactose metabolism. So what enzymes are those? Like Z, the first enzyme would produce the beta-galactosidase enzyme. Now this is an enzyme that is needed for breakdown of lactose because it's a disaccharide. So when you break it down, it will produce glucose and galactose that will be utilized as a source of carbon. Now I'll come to the other function just in a moment. The next enzyme by Lac Y gene is the lactose permease. So there has to be some system that allows the lactose molecule to enter the bacteria, right? And lactose permease allows the lactose to enter the cell. So it will facilitate the transport of lactose into the cell. So there has to be something that brings the lactose in, that is the permease, and something that breaks it down, that is the galactosidase. Now the Lac A is the gene that codes for transacetylase. Now what happens when this lot of lactose is there and cell is transporting a lot of lactose inside, the level of beta galactosides would rise in the cell very much. And if it goes to a particular level, it can be, the accumulation can be toxic for the cell. So the work of this transacetylase enzyme is to acetylate the glycosides that is coming lots and lots of quantity in the cell and allowing them to escape from the cell. Just simply throw it out of the cell so that it does not accumulate up to toxic level. Okay, that is the function of three enzymes. Now what happens in a normal condition when a bacteria is present in an environment? Generally there will be preferable sources, simple sugar like glucose will be available for the cell to use. So in that case, the lactose metabolizing genes are not needed. So in a normal condition, actually this operon is in off condition. That means the transcription of these genes are inhibited. So now let's see how that happens. This is a general normal condition, okay? So this is where our inhibitor gene comes into picture. This is an inhibitor gene. That means it produces an inhibitor or what we will call a repressor. Now this gene I is a constitutive gene. That means it is always expressed. So you will always have repressor at any given point of time in the cell. In absence of lactose, this repressor, as the name says, it has to repress, it has to inhibit the transcription. And how it does so? By binding with operator. Okay, that is what is operator, function of operator, Lac O. That it has a site where the repressor will bind. Now this operator actually overlaps the promoter a little bit, okay? So this repressor will come and bind to this operator. Now what happens? Promoter is the region where the RNA polymerase will come and bind, right? Because it can recognize the promoter sequence. Now if RNA polymerase comes now, it will, it definitely will try and come and bind to this promoter site. It cannot actually bind to this promoter because it cannot access this promoter completely because of the repressor. And as a result, it cannot pass through the operator and it cannot transcribe the gene. So there will be no transcription. This is the natural normal condition. When there is no lactose in the environment, the inhibitory gene would produce a repressor that goes and binds to the operator. As a result, operator overlaps the promoter a little bit. So RNA polymerase cannot access the promoter unless it can bind to the promoter and there is open place where it can pass through the operator. It cannot transcribe the gene, right? So RNA polymerase cannot bind to the promoter, and as a result, there will be no transcription, no enzymes produced, no problem. Cell is saving all the energy. This is the normal condition, which is the off mode. Off mode of what? Off mode of gene transcription. Now let's say, for example, the same bacterial cell is now in an environment where there is absolutely no glucose. and there's only lactose present. So now cell doesn't have any other choice. It has to utilize or metabolize the lactose. In this case, what happens is, if you look at this, what do you think should happen, logically? It is the repressor that is causing the problem, right? That is going and sitting over at the operator, and the RNA polymerase cannot transcribe the genes. So that means the repressor is a problem, and we don't want the repressor to bind. So that can be taken care by the presence of lactose itself. What happens is, if suppose in the environment lactose is present, that lactose, some molecule of lactose, would be taken up by the bacterial cell. And this lactose would be converted into an isomer of lactose, that is the allolactose. And this conversion is by beta-galactosidase enzyme itself. I said beta-galactosidase has two functions. One is to break down the lactose into glucose and galactose. The other function, it will cause the isomerization of lactose into allolactose, because allolactose is the actual inducer over here. It is the allolactose that can bind to the repressor. Now this repressor over here has actually two binding sites, okay? One is the site which binds to the Lac operator, and there is another allosteric site where the inducer binds, okay? And inducer in this case is the allolactose. This allolactose will now bind to the repressor. Now the moment allolactose will bind to the repressor, it causes the conformational change. The shape will change in a way where it can no more bind to operator site. So it will fall off, okay? This will fall off. As a result, what will happen now? Now the promoter site is free to be bound by RNA polymerase, and it can easily pass through the operator and will start synthesizing all these three enzymes: beta-galactosidase, lactose permease, and transacetylase. As the genes are expressed, there will be one single polycistronic mRNA, mRNA that will be produced. Now it's a bacteria, right? So there will be polycistronic mRNA. What does this mean is, it has all the sequence of three enzymes together, and that will be transcribed into three individual enzymes. So this is what happens when there is lactose present in the environment. The genes get switched on. Simple enough to understand. Without the lactose, the inhibitory gene produces the repressor, which binds to operator. As a result, RNA polymerase cannot access the promoter, cannot pass through the operator, and the genes will not be expressed. If there is no glucose present in the environment and only lactose is present, cell has no other way but to utilize or metabolize lactose. In that case, the lactose molecule will be allowed to pass in the cell by permease, and it will be converted into allolactose by beta-galactosidase, and this allolactose is the inducer, which can go and bind to repressor, resulting into conformational change. The shape of the repressor changes in a way that it will fall off from the operator. Now the operator site is open, polymerase can simply transcribe all the genes, enzymes will be made, and it will start metabolizing lactose. Now there are a couple of questions that should come in our mind when we are looking at this. What I am saying is, when there is lactose present, some molecules should enter in the cell by lactose permease, and few lactose would be converted into allolactose, the isomer, by beta-galactosidase. At the same time, I am saying these are the these enzymes are the product of Lac Operon. So, at the time when lactose is present, repressor is bound. How is it possible that already beta-galactosidase and permease is present in the cell to allow these molecules to enter and convert it in the isomer? Right? How does this happen? Now this is called the lactose paradox. So what happens is, for any repressor or such protein that is bound to the DNA, there will be a time that it might just fall off from the operator. Okay, it is not like it is bound all the time, but there can be just a gap of a fraction of a moment where the repressor fall off from the operator, and RNA polymerase will take advantage of that, just a second, and will pass through this operator, transcribing these genes. Okay, and immediately the repressor will again come back and bind to the operator. So it might happen that sometimes, and it happens, sometimes the repressor does fall off and RNA polymerase takes advantage of that fraction of a moment, transcribing these enzymes in very, very low quantity. Okay, it's very, very like the baseline level of beta galactosidase and permease is always present in the cell. At any given point of time, very, very low quantity of these enzymes will be there. So what happens is, this permease will actually allow the lactose molecule to enter in the cell. Like, the study has been done where they have seen somewhat around 300 molecules of permease present at any given time in the cell. And very few molecules of beta galactosidase that are present will actually convert this lactose in the allolactose, so that it can go and bind to repressor. And once it does, once the repressor is removed, then you will suddenly see that there will be like a huge improvement in the levels of this galactosidase and permease enzyme in the cell. Okay, that's how the lactose actually enters, and that's how the allolactose is actually formed. Now this is just an extra bit of information which is called the lactose paradox. Just if you are wondering, how without Lac Operon being activated, before that only how these enzymes are present in the cell. So this is the reason. Now one more question that comes in my mind is, why can't directly lactose go and bind to the repressor? Why it has to be converted into allolactose? Now I did not find the exact reason or something which could convince me completely, but what I found is, it is to avoid the unnecessary gene expression if there is only very low amount of lactose present in the medium. That means only if there is a good amount of lactose that is present and a good amount of molecules of lactose that are coming in the medium, that would be converted into allolactose. So if the repressor gets bound by the allolactose, it's a kind of a signal that there is good amount of lactose. Go ahead, transcribe the gene, produce the protein, and metabolize it. But I'm not 100% convinced with this. This is what I found. If at all you guys know why only allolactose is needed for binding with the repressor, why not directly lactose go and bind to the repressor, please comment below down and let me know. So that's all about the Lac Operon. By looking at this now, I think you will know why it is called negatively controlled inducible operon, right? So now next video, this was the negative control of Lac Operon. Next video we will continue with the positive control of Lac Operon. Like what happens in the cell if there is, like I said, the scenario glucose is not present and only lactose is present. There can be a situation where both lactose and glucose will be present, right? In that case, even though lactose is present, the cell has to be smart enough to switch off the genes and utilize the glucose first. And once the glucose is depleted, it should again go back and switch on to utilize the lactose. So that is a kind of positive control which we will continue in the next video. So that's all for now. That's all about Lac Operon, about the negative control. And I hope this video was helpful. Do subscribe to the channel for new video every week, and I will see you next time. Until then, keep learning.